Okay, gram staining. Gram staining is a known as gram stain or gram matches, which is used to differentiate or classify bacteria species into the two large groups, which is gram-positive bacteria and gram-negative bacteria. The purpose of doing this gram stain is usually to identify or classify the bacteria. So there are four reagents that we use in this crime stain, which is crystal violet, granitidines, alcohol, and safranin. So now we'll look at the steps. Okay, here is the steps for crime staining. Heat fixations, put some crystal violets, rinse, and then put drops of iodines, rinse with water, and then colorize with alcohol, flood the slide with the safranins, and we are going to view slide under the microscope. So now we're going to start. First, heat the inner coating loops to remove the impurities or contaminants on the loop to avoid the contaminations until it's become a redness on it. So let water dry on the slides. Now we are going to start the process we call heat fixations. So heat fixations where we heat fix the slide. So now turn on the Bunsen burner and pass the slide around 2 or 3 times, about 1 to 2 seconds. And then we're going to start with the first stain or initial staining solution, which is crystal violet. Let this liquid stand for a few minutes and rinse out with distilled water to remove the excess crystal violet. Then we're going to add iodines. Iodine here acts as fixations, where the iodine fixes the crystal violet into the cell wall of the bacteria by working as mortar. Mortar is a more likely substance that increases the affinities of the cell wall for stain by binding to the primary stain. So then remove the excess iodine with rinse out using a distilled water, and then we're going to apply the alcohol. Alcohol here acts as a decolorizer. This alcohol decolorize the color of the samples. The alcohol dissolves lipids found in the outer cell membranes of gram-negative bacteria, allowing the crystal violet iodine complex to leak out the thinner bacterioglycan. Once you The complex that we call crystal violet iodine complex is came after the crystal violet and iodine same process. And then we use out the excess alcohol by using this water. So the alcohol that we use here is the ethyl alcohol or known as ethanol. So then the last standing solution is known as safranin. Safranin is also used as counter staining gram staining. In this test, the safranin directly stain the bacteria that has been decolorized. And then we remove the excess safranin by adding distilled water. Okay. With safranin staining, the gram-negative can be easily distinguished from the gram-positive bacteria. So then we view the samples under the microscope. Ayun. First of all, we will heat the inner coating loops until it's become redness to remove the impurities and the contaminants to avoid the contamination. So let the water dry on the slide, then we're going to start the heat fixation process. Okay, heat fix the slides. By just pass the slide, first you click on the Bunsen burner and just pass the slides around two or three times, about 1 to 2 seconds. So the first initial staining solution, which is crystalline, is entered, and that liquid stand for 1 minute. After that, rinse out with the distilled water to remove the excess crystal violet. So then we're going to add iodine, a few drop iodines. Iodine here acts as a fixations, which where the iodine fixes the crystal violet into the cell wall of the bacteria by working as mordant. Mordant is more likely the substance that increases the affinity of the cell wall for stain by binding to the primer stain. Primer stain here is known as a crystal violet. Then we remove the excess ions by using a distilled water. Then we're going to add alcohol. Alcohol is act as a decolorizer, where the decolorize the color of sample. The alcohol dissolves the lipids found in the outer cell membrane of gram-negative bacteria, allowing the allowing the crystal violet iodine complex to leak out the thinner peptidoglycan. Crystal violet iodine complex is formed once the crystal violet and iodines staining is done. So, and then remove the excess alcohol by using a distilled water. The alcohol that we use here is ethyl alcohol, or known as ethanol. Then the last staining solution is also known as so the last staining solution, which is safranin. Safranin used as a counter stain in gram staining. In gram staining, safranin directly stain the bacteria that has been decolorized. So then we rinse out the excess safranins by using a distilled water. With safranin, the gram negative can be easily distinguished from the gram positive bacteria. So we are going to view the sample under a microscope. Okay, here is the samples. Here is the viewing our samples under the microscope. So there are two types of bacteria. Seems like there are two types of bacteria here. So uh, one is gram positive bacteria and gram negative bacteria. So the 1 gram bacteria, gram positive bacteria is in bacillus shapes. Gram negative bacteria is in bacillus shapes, and another one bacteria, a gram positive, another types of bacteria, which is gram positive bacteria in coccus shapes. So we're going to examine this example. So as we see before, the bacillus shaped bacteria is a gram negative bacteria. So here, this is okay. Okay, these types of bacteria might be in our sample, which is Pseudomonas aeruginosa. While this is for the gram-negative bacteria, and while for gram-positive bacteria, it might be Staphylococcus aureus, which is in a coccus shape.